right now. So Mars is at one of its closest approaches to Earth right now, and tonight's clear. I think tonight's going to be clear. I'm going to go out and look for it. Also, in our astronomy notes, I think tonight, I think. Sorry, checking my smartphone. Yeah, at 10 p.m. tonight, the ISS is going to be coming over at 87 degrees, which means 90 degrees is like right overhead. 87 degrees is nearly right overhead. It's coming at 10:01, so it'll probably be right over your head at about 10:03. If you go out tonight and you want to see the ISS, I will still accept, if you get a video of it on your smartphone, I will go back and give you bonus marks on the first astronomy, the chapter 10 and 11 test. It's up to you. Let's continue on. Pause here. No, we're going to continue here. Checkpoints. So what do checkpoints do? Checkpoints are biological, chemical systems that prevent the cell from dividing if conditions are poor. What do I mean by poor conditions? Number one, not enough nutrients. That's why people get skinnier when they're starving. Cells are dying off, new cells are not replacing them. Oh, yeah. Okay. Number two, if the DNA is not fully replicated, the last thing you want is a cell with the wrong copy of DNA. In fact, that's what radiation does. One of the reasons radiation can cause cancer, radiation bombards your DNA and it can knock electrons off of the atoms in your DNA, making them charged, and like charges repel, unlike charges attract. They'll combine in the wrong way. Those base pairs will combine in the wrong way. The C, the G, the T, and the A will combine in the wrong way, and now that DNA gets replicated and passed on. That's why radiation is bad. Or if the DNA is damaged. The three of these, especially the bottom two, happen fairly often. And your body is pretty good at then saying, okay, we're not going to prevent the cells from dividing. In fact, it's going to kill that cell off. So there's kind of here during interphase and during mitosis, here's that little circle I showed you. There's the fork in the road for mitosis. It goes like this. So interphase starts, here's growth and preparation. Stop, the cell lacks nutrients to support its growth, or the DNA is damaged, the cell will be destroyed. A checkpoint will come along, that cell will not reproduce. Then the DNA replicates, starts to form chromosomes. Stop, the DNA has not replicated correctly, or stop, the DNA is damaged, the cell will be destroyed. It will not reproduce. And then during the mitosis procedure, stop. Some of the chromosomes have not attached themselves to spindle fibers in metaphase. They're not aligned properly in the middle. Or in anaphase, they haven't all moved away apart. Some of the chromosomes did not split up. That cell will be destroyed. If all goes well. That's the hope. That's the plan. If this doesn't occur, cancer is when the cell cycle goes wrong. Okay. This is something I'm particularly aware of. The cancer gene runs through my family. Most of my uncles, although they made it to their 70s, died of cancer. My dad has battled cancer. So some of it's genetic, some of it is also environmental. So what's the official definition? Cells that divide when they shouldn't because the checkpoints stopped working. Okay? Cells that divide when they shouldn't because the checkpoints stopped working. Normal cells stop growing before they get too crowded. But cancerous cells don't do that. They keep growing, resulting in what do you call a mass of cancer cells? Begins with the letter T, a tumor. So this all comes out of the cell cycle. Cancer's really tricky. While there are often common factors in different cancers, each cancer is fairly unique because they contain, each of them, your own unique DNA. Two people with the same type of cancer may have different mutations. They may have very different DNA changes or DNA damages, making their cancer different from each other. Some risks for cancer are hereditary, they're part of your genes. Some are environmental. There are lifestyle choices, there are lifestyle behaviors, there are environmental factors that can increase your risk of cancer. So here's a few. Let me zoom in a little bit. Okay. Alcohol, linked to cancers of the mouth, the larynx, the throat, the esophagus, and the liver. Food, one of the things we're learning is obesity increases the risk of many cancers. And salt-cured, smoked, and nitrite-cured foods are linked to cancer of the stomach and the esophagus. Some additives and some preservatives are linked to some cancers. Now, by linked, we say there seems to be a connection. Cancer is a really tough one to nail down. It's not very often that we can say definitively for sure, hey, that absolutely causes cancer. Even smoking was a really tough one to pin down, which is why the smoking companies, the tobacco companies, were able to block that for so long. High-fat diets may contribute to the development of some cancers. Home. Exposure to certain materials from manufacturers, such as carpets, plastic, rayon, insulation, some cleaners, some toxic chemicals, paints and other chemicals. Radiation, X-rays, this is why we minimize X-rays. The sun, this is why we tell you to wear sunscreen. Radon in cigarettes. Did I put this chart in your notes or not? Okay. Sunlight, exposure to sunlight is considered a major cause of skin cancer. Wear sunscreen. Tobacco. So tobacco is responsible for about 30% of all cancer-related deaths. Don't smoke. It's responsible for most cases of lung cancer. It's a cause of mouth cancer and throat cancer in chewing tobacco. And then where you work. Some jobs, you're around chemicals. So you may see commercials, for example, people that used to work in asbestos mines inhaled tiny asbestos fibers. They got into their lungs and they're at risk for developing lung cancer. Some of these you know. I think a good test question would be list some causes of cancer. So you may, in your notes right now at the bottom of the page, might even want to jot down some causes of cancer. Sunlight, if you want to be even more fussy, UV rays. Tobacco, alcohol, obesity. Those are ones that are pretty easy to remember, that probably some of them you knew already. Okay. So, what was that? Well, not just that. A lot of pets, especially certain species of dogs, are very susceptible to cancer because they're so inbred. Purebreds are beautiful dogs. We're really doing them a genetic disservice, though, because we're breeding in common either like risks to. I told you I have a genetic risk of cancer running through my family. Well, a lot of purebreds have that really going through. So really, mongrels, mixed breeds for dogs, much better in terms of their health. Yeah. Yep. Are we going to find a cure for cancer? I want to think so, but we're finding that a single cure, because each cancer is so unique, and even if it's the same type of cancer, Mason, I don't want to personalize it. Patient A might have liver cancer, and patient B might have liver cancer, and patient A responds wonderfully to a treatment where patient B's liver cancer doesn't respond at all to the same treatment. It's really a tough one to nail down. So, is one cure for all cancer likely? Does that mean that cancer research is pointless? Of course, absolutely not. Because what we're getting better and better at is curing specific types of cancer, and we're just on the threshold now, it sounds like, of being able to create cancer treatments customized to your particular DNA. We're just starting, for example, to be able to analyze someone's DNA and say, oh, that person probably won't respond to this drug or this medication, but they'll respond very well to this. Up until now, it's really been, Brady, trial and error. Try this. No. Try this. No. Try this. And our success rate is hugely better than it was 50 years ago, but it still sucks. Still sucks. So here's a picture from page 161. If you want to, you can look at it in your books. I'll make it bigger here. This is what we call metastasizing. That's the fancy word for cancer spreading. So I got to make this a little bit smaller, Mr. Dewick. There we go. So here is a blood vessel. This is where the nutrient. Let me make my eraser so you can see what I'm pointing at. Here is a blood vessel. Here's where the nutrients are coming. Here is a lung cell. Here is some lung tissue. Normal lung cells are right here. A couple of cancer cells have formed. They got past the checkpoints. They're going to start to draw nutrients from the bloodstream as well, and so you're going to start to develop a tumor. This would be a tumor on your lungs. Metastasizing is when the tumor gets big enough that a few cells make it into your bloodstream and break off, and this is how cancer spreads. And this is why you'll hear us talk about early detection being very, very important. I just saw last week that a university had come up with a procedure for detecting lung cancer much, much earlier, because typically lung cancer only shows up when you're coughing, when you're not feeling well, and you go for an x-ray and they find a spot on your lung. So they're trying to find, is there something you could breathe into, and could that somehow detect that you've got lung cancer well before it becomes big enough to spot on an x-ray? Then we would add that to a yearly physical. That'd be part of your, every year you blow into a tube and it can detect certain cancer chemicals in your coming out of your lungs. Maybe that could early detection. You want to get it before it metastasizes. Then that would be susceptible. That would be responsive to surgery. Oh, didn't want to do that one, Mr. Dewick. Too small. So this process is known as metastasizing, cancer spreading. That's probably a good word, vocabulary word that I might ask you. Oh no, I have it in your notes. So here we go. Metastasizing, like it sounds. metastasizing. Metastasizing. This is when cancer cells break off from the original tumor and travel to other sites in the body through the bloodstream. It's much, 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 much tougher to treat cancer after this happens.